keynote address. Partnership and collaboration and support we're talking about today. I think this is going to be an amazing talk that's going to totally cover all of those things. I'm really excited today to have Hardeep Phull with us. He is a Bravo and UBRP alumnus, here from 2000 to 2003, around that range. Yes, and he will definitely explain more about that. He was also involved in our Bravo program and was able to conduct research in Poland as part of his experiences there. And we are also fortunate to have with us today Mike DeHolos, who is one of his former UBRP mentors. He actually has Georgina Lambert here as well, from like 22 years ago, right? Super awesome. So I'm going to ask Mike to come up at this point, who's here all the way from the University of British Columbia, to introduce Hardeep, our speaker for the keynote talk. So,
thank you, Jennifer. Thank you, everyone, for being here so early on Saturday morning. It's my pleasure to introduce Dr. Phull, Hardeep, as we called him 23 years ago when I was a postdoc in David Galbraith's lab with Georgina Lambert, the lab manager who held everything together. Hardeep will tell you more about his professional journey in a couple of minutes, but I'm going to summarize it here as much as I can summarize an 18-page CV. After graduating magna cum laude from the University of Arizona with a Bachelor of Science, he completed a medical doctorate with special qualifications in biomedical research from the Cleveland Clinic, and a research fellowship at Stanford, an internship, residency, and Master of Science degree at Scripps. And then he became a fellow and was selected as chief fellow in hematology and oncology at the University of California, San Diego. And he has been a clinical faculty member there at UCSD since 2018 and is currently director of oncology at Palomar Health, which I think is California's largest health organization. And in addition to his very busy clinical practice, which benefits hundreds of patients, he is an expert reviewer. He has research which is still being cited. He is a consultant, a columnist, a podcaster, board member, industry thought leader, a mentor, and a patient advocate, with too many awards to list here. So obviously, Hardeep is pretty special, right? No. No, he's not. And that's the message I would like you to remember today. He's not special. And why do I say that? Well, if you met Hardeep 23 years ago, you would absolutely not say, oh, there he is, future head of oncology at Palomar Health. Future Scripps, Stanford, and USD alumnus. So, in fact, Georgina, we kind of made fun of him, didn't we?
We still do.
Okay, so I emphasize this because the thing I've learned in more than 25 years of mentoring students is that one of the most difficult ideas that many of you need to overcome is the idea that you need to be special to do great things. To be clear, Hardeep is a great human being. He definitely has a lot of innate abilities and natural talents and personal virtues. But as you'll see in his presentation, he became a successful person, the person who he is through effort, resilience, experience, and a willingness to take risks and seek opportunities. And I'll highlight particularly, for example, he knew he was always interested in healthcare, but he didn't hesitate to take an opportunity to work in our lab in plant genomics. So again, the message to the students is you might be doubting your own innate abilities. You might be thinking, well, I'm not good enough at this to be that. And I think both of us are going to give you the message, knock it off. Okay? Your innate abilities are probably sufficient, and the fact that you're here doing something like UBRP shows that you're on the right track, you're doing the right things, seeking opportunities and experiences. So I will turn it over to Hardeep in a minute, but one very quick story before I close. I said we made gentle fun of the future
It wasn't gentle.
Dr. Phull, but it really went both ways. He and another undergraduate student wrote a song about me. My first name is Mike, and at the time there was a song, Be Like Mike, which was about Michael Jordan. They wrote a tribute to me and posted the lyrics around the department.
You're welcome.
And so I jokingly wrote Hardeep minus 2 hours of pay on the bottom of one of those and left it on his lab bench. I found out at dinner last night, Hardeep, being the person of great integrity that he is, actually took 2 hours off of his timesheet that week. This is the first I've heard of it. So I will invite Hardeep, Dr. Phull, up to the podium to present his talk and to receive $7, which was... I know, but we only had $7 cash, so I'll owe you the other $7. But I do have Canadian quarters as interest, so you can use those. You'll use them and come visit me. All right, so please join me in welcoming Dr. Phull.
Wow, what an introduction that was. I was not expecting that. But as you'll see, we're talking about science, medicine, research, but really life. And if you see the title of my talk, and what Mike said, it's about getting out of your own way. And if you do that, you'll see that in your spot, you have more potential than I ever had. In fact, when I was in your spot 23 years ago at this very conference presenting here, not in this building, I didn't think that. I was in the back there, looking at my notes, pretty nervous about posture. I'll get you through that. We'll get through this. So Bravo, as you know, this is taken from the website. The V in Bravo stands for Vista, and I thought about what that meant. It means openness, and keeping an open mind and sights. As you look at this talk, don't think about me and what I've done. Think about your journey, where you're on your path, and where you're headed, because this is really about you. So lesson one, this is really lessons to myself, but feel free to take some if you want. You got to start somewhere, and what a great start this morning. And so this is Jelenia Góra in Poland. A lot of my pictures today will be from Poland for my Bravo trip. But just remember, you're exactly where you need to be. There are no mistakes. The only mistake is not making the most of where you're at. So with that, I'll talk about what I did in the plant sciences lab with David Galbraith under Mike DeHillos and Georgina's mentorship. We were making these really cool glass DNA microarrays. 40,000 genes could be printed on these glass slides and then tested for genomic expression. And we were doing this all in plants, of course. And all of this was done via automation with robots before there was any thoughts of AI, before there was any social media. So we were a pretty hotshot lab internationally, and what a great place to learn all of that. To give you an idea, you use this technology by labeling DNA from your target organism of interest. It could be a cancer patient, it could be a plant. And you basically expose this organism to different stressors, and you look at the DNA that's labeled, and you hybridize it to these DNA molecules of the entire genome on glass slides, and then look at differential expressions. So for example, in this cancer patient, let's say a normal one versus an abnormal one, you could look at a green spot, and you could say that was a higher expression of a certain gene in this cancer patient who did well versus one who did not. It can formulate hypotheses for saying that could be in the pathway for a treatment for this disease. Same with plants. You could say drought-resistant plants do this better. Let's engineer these genes into them so that we have better food supply and agriculture and yield and so forth. So here's that printer, the automated, cool robot. I mean, amazing thing to work on this thing in our lab. And so my small project, looking back, I actually reread my thesis and I was just like, what are you doing? But looking back, I figured out what I was trying to do. Basically, we were trying to take DNA molecules, instead of doing these long strands, which are finicky and take a long time to synthesize, we said, let's just make oligonucleotides, 20 to 30 bases in length and print those. But they're very finicky. They don't bind to the slide, and we didn't know the right way to get them on there. So my project was really looking at the buffer concentrations and various variables, the concentrations of the probes, and then looking at data analysis, including how good the signal was doing this approach, how good the variation and the standard deviation. I learned a ton of statistics, which I would use later in my career as a clinical scientist, doing just that. So basically what I ended up finding, and by the way, these are just Microsoft Excel files. You don't need to have a fancy poster or anything to learn science and to learn patterns and to make a difference. But what I down to a 60 to 80 micron or micromolar of primer, and a certain buffer combination would work best to produce the best signal with the least variability, the least noise. And I basically showed that after various experiments, immobilizing slides and doing different techniques in microarrays. Pretty simple project now looking back in hindsight. I'm like, I can do that in the afternoon. But actually that took about a year alone. And it was very satisfying to go through the thought processes, even today when I reread them and realized what I was doing. So my very first publication was at this conference, and I still say that was what started me and gave me pride. And all of you today should add this to your resume on your project. That's how much that means, and that's how much it'll get you somewhere. So, I talked about this, or he did, right? This being this joker, and I really mitigated down my humor and talk today, but this is who I was. Look at this guy. I mean, this is who you have to deal with. So, we talk about taking risks, but someone has to take a risk on you. And Mike, Deolis, Georgina, and the rest of the lab took a major risk on a guy who's acting like this. Now, this is all closed and it's all for fun, but this is before selfies, so that's all I can do with a digital camera. But I actually did know what I was doing, and the fact that I was doing something so advanced meant that all these international collaborators coming to our lab would look to me for advice. An undergraduate, a sophomore, a junior. I look back and think I wouldn't even trust a medical student with this stuff. And that's the arena I was in. What a privileged place to learn. And so I learned to also be a lab ambassador, and I got to help people who were traveling and bring them to our home. My mom was in the front audience, and she would always, I would call her, I'm bringing over so-and-so and have dinner ready. And it was never, we'll see about it. It was, all right, let me know when. And we went to Mount Lemmon. We went traveling to the Grand Canyon. I felt like an ambassador. People asked, how did you have time for that, and why did you do that? And I think it has to do with upbringing. I think both my mom and dad both did that for other people in our community. And so I learned very quickly, like, that's the right way to treat people. And this paid it forward, because later on, you'll see how many people did that for me, and it was so helpful. So, lesson 1, start somewhere. Let's talk about Bravo. And this is about speaking the world language, because I was basically isolated in Arizona in the US. This was the front picture of my website I made during that time. Again, this is before social media or anything, so I just made a little online journal, because we had to make one anyways, Carol said, and I said I might as well make it public. And I had pictures of there and what I was doing, and a little map that was interactive. I wasn't a web programmer, I just picked it up. But I thought that was cool, and I still have this website up just with no pictures. It just has all of my ramblings of what I remember and what I did every day. This is what I learned from this experience, is you cannot develop universal truth or compassion until you see or speak the world through the eyes and tongue of someone else. And I still use that in my practice today. So Poland, as a country between Germany and many other countries, and it's positioned right by the Baltic Sea, I was in this city, Bydgoszcz, which I still couldn't pronounce for many, many years afterwards. But going there, we learned how to use the Z's and pronounce them, and I got a lot of mentorship in that. There were many people who were not English speaking, so I had to really learn and get out of my comfort zone. So these are sugar beets. So I now transitioned from certain plants in the lab to sugar beets. And there were massive fields of them all over Europe, and I thought, why sugar beets? Well, it turned out in the Napoleonic Wars, England had cut off sugarcane production, and so people began to rely more on sugar beets. There's still 20% of the world's sugar production, which I learned only a few weeks ago. It used to be a lot higher in the European Union, like 50%. The next question I get asked is, how does this taste like if you pull it out? And I say, well, it tastes like dirt and it's really bitter. So you have to wash it and you have to boil it, and then you extract the sugar. So my project with Elvira Selinska and Ivana Gondrajczyk, who both visited our lab, by the way, and were one of these people who I worked with so closely, that led to this collaboration. Had I not had that experience, I would never be in Poland. And so they mentored me, and we talked about making a sugar beet DNA microarray. And so these are slides from my actual talk from back then. I can't believe I made these funny animations and beats. But basically there was a strain that made a big yield, and then there was a strain called Z, which made a very, very sweet variety. And the thought was to figure out what they're doing differently in the genome, to figure out what are those genes and why, and then to study that, and then to obviously try to make genetically modified organisms that can do the best of both. So there's a lot of steps in this, and so I initially got very caught up in the steps and how to do what and how to do things. I actually made this DNA microarray like animation back then for the students to say how I was hybridizing two different labeled probes. But it was a very complicated project, as I told Mike, for an undergraduate, because you had to make a full cDNA library, including insertion of mRNA extraction, cDNA probes, and insertion into vectors, propagating that. A very ambitious project even to do here, much less abroad. And so the other problem was trying to communicate any of this, especially to people who did not speak English. So I found slides in Polish, and at one point I knew how to read this. I don't at the moment, I'd like to apologize. But teaching that in a different language made me learn it even better. I had to first learn it in English, of course. So this is me lecturing at the university, Ate Ere, it was called. And you can see the mistake. I looked at the slide and I knew exactly what I was doing wrong. I'm looking away from the audience, right? But that's me still learning the slide, believe it or not, at the presentation. So much has happened since then. And as you do your presentations, remember to make eye contact and talk to your audience. The data will speak for itself. So this is the university. It was Akademia Technicko-Rolnicka. I think I pronounced that, or Ate Ere. And this is me in the lab, hunkering down. This was between the fall and the winter, and snow was coming down. My first snowfall outside of Mount Lemmon, by the way. So it was an interesting time. But I got stuck in the precipitation that wasn't just the winter, just in the lab. I couldn't get my project and the DNA strands to be concentrated enough to do cDNA libraries. And I didn't know anyone there except in my lab, but my lab knew some other people and said, you should go reach out to this person. And I didn't know any of these people, but I emailed them, and we didn't have cell phones or text, and you'd be shocked. This university, which was nearby, Wilkie University, Yaroslav, and Magdalena, reached out and invited me to use their equipment to help me precipitate this DNA out because we didn't have the equipment. So here's this 20-year-old in a bus in Poland with this foam box with DNA in it. They didn't know there was DNA and samples in there. On a bus, going to another lab, and everyone's staring at me like, what is he going to do next? Of course, I was going to their lab, but they welcomed me with open arms. They had coffee waiting. I mean, I remember feeling like a diplomat, not a scientist. I don't think they even knew I was an undergrad at that point, the way they were treating me. But it was such amazing collaboration from basically complete strangers. And I just recently I forgot their names, so I recently found them online and what they're doing, and it's amazing. It led to a whole discovery of all the projects they're doing, and it just brought back that moment of how much gratitude and kindness was given to me. And so, of course, joy of friendship and collaboration was everywhere, including in our own lab. This is Elvira, who I'm standing with, and then Ivana and Dorota, and they helped me troubleshoot these things, and we worked together. So science is a language. You work together and speak it together. I won't go into this much, but this is why I failed my project, and it had to do with a kit that was shipped to customs in Warsaw and sat there too long at 4 degrees. A failure in a sense, but I recognized it. I was able to troubleshoot that, and I was able to call this company's strategy and figure out what was going on, and realize that industry is a partnership, and it wasn't anyone's fault, but figuring out the problem. So this was my final, my talk that I gave for Bravo to an audience like this about my experience, and I talked about isolating Z's and cDNA, but really what I thought about was this flag. I kept saying this is Warsaw, this is Warsaw. This was actually in Toruń, Poland, and that's where Copernicus basically was, and this flag was right off of Copernicus's tower. So what I realized was this was like my breakthrough Copernicus moment, that he made a rebellious idea that the sun doesn't revolve around the earth revolves around the sun and all the other planets, and that's the heliocentric theory. But it made me realize when I saw that flag and I saw this power of this statement and his rebellious nature to be a scientist and pursue truth, that the greatest truth is to realize that the world doesn't revolve around us. We work together, revolve on the same planet together, and there's interconnectedness, and we're all on the same journey together. So besides that, I couldn't do this talk without probably doing hours of just pictures, but certainly what a beautiful cultural history in Poland, and I got to travel the entire country, all by train, actually. What I'll tell you is the Polish people are warm, kind. As my mom experienced, she also visited me. They're in a place in history where they were invaded and were in a geographic region where it was always destruction and rebuild, destruction and rebuild. And some of these buildings up here, you see, look authentic, but they were actually only 50 to 100 years old because they had to be rebuilt after World War II. Amazing that a country like that can thrive and still have so much positivity in its people, despite all that destruction. And of course, the biggest destruction I saw was in Auschwitz, this concentration camp. And so, still affects me emotionally because I look at this picture and I remember feeling numb that day, shell-shocked, in catatonic state, because it was so serene and beautiful. It just sounds silly. When I was there was birds chirping, there was a beautiful breeze. It was one of the best days of September in Poland I'd ever been in. And yet I saw this, and I saw what had happened there. This is the biggest concentration camp in Europe, and 1 million people died here. Just think about that. My daughter recently saw this picture and said, "Oh my gosh, you were seeing people dying in Poland?" I said, "No, it's just a shell of it." And so it's interesting, Carol Bender actually had me reread a passage from my time there, and this is what I wrote. I wrote, "We see the shell of what remains. We haven't lived here. We haven't died here." It's just a powerful statement of how much history is all around us if we appreciate it and if we can learn from these lessons. But of course, what matters most is people, right? And these are all the wonderful people. Remember that slide I talked about being lab ambassador? Well, these were also lab ambassadors who gave so generously of themselves. Ivana, who was pregnant at the time, that was her first baby. You can see her holding her stomach there, but we went through that together. I felt like that was a big part of our life that I got to be a part of. We had a lot of parties, we got to travel. They even took my mom out for dinner, and still the best Indian restaurant I ever had in Warsaw, Poland is that picture right there in the top right. A beautiful, beautiful cultural experience with wonderful people. And I went viral, if you want to call it that. There was only Google back then. There was no Twitter or Instagram. So this picture, when you used to search my name, this is what came up. Or if you searched Bravo or Uber, this picture. I wasn't even smiling properly, but people would ask me, what are you doing? And we didn't have Photoshop back then, but all my friends used Microsoft Paint and definitely embellished this picture way more than I wanted. But I also recognized only yesterday that I was going viral in Poland, and Carol showed me this. I didn't even know this existed. They published a whole article on me in their internal bulletin, and that's me in the lab, and that's us at this lab party. I just was so touched. I didn't even know this existed until yesterday, Carol. And so you don't need to go viral. You're already viral, and whatever it is, it doesn't require followers or anything. It requires just being present and making a difference. So, speaking of the world's universal language is lesson number 2 for me, definitely. Lesson 3, this is cool stuff. And this is me transcending artificial boundaries on my quest to be a pilot, which was always a dream of mine, and something really enabled because I was in